So, we carry our phones with us almost everywhere we go, and at this point they're almost an extension of ourselves. So that when we want to be able to do something new, we just download an app, and suddenly within seconds we can navigate a city, join a new social network, or be able to identify a song while it's playing. So my question is, if we live in a world where it's so easy to download new capabilities, just like Renee said, why can't we do that with our biology as well? This is a question I've been obsessed with since I was 12, and for the past decade, I've been trying to develop both tools and programs towards this mission. But I learned a very difficult truth. I just learned that the technical tools we have are simply not good enough. What does that mean? So today, when we talk about bioengineering, we talk about genome engineering, technologies like CRISPR, where we go and edit our cells' own DNA. This is extremely useful when the problem lies with one of the 20,000 genes we have in our genome. However, that only accounts for 3% of global disease burden. So what are we going to do with the rest? Neurodegenerative disease, hematopoietic, muscular diseases. How are we going to solve those? They're a result of dysregulation between tens and even hundreds of genes, or potentially unrelated to our genome altogether. So there really isn't a single target to aim at. What we need is an interface where we can sense what are those higher level states or behaviors of our cells and learn to modulate them. Well, luckily, evolution has already created such an interface for us. Let me explain what I mean. So, the first life forms on this earth, they were tiny, microscopic, single-celled, and invisible to the naked eye. But then, about 1 billion years ago, something happened. One of our ancestors swallowed another cell that had the ability to generate energy using oxygen. And usually that would be the end of the story, but in this case, something else happened, and they integrated. And now, 1 billion years later, we call that swallowed cell our mitochondria. And the result of this merger was not just a cell with more energy. It was a completely new cellular infrastructure. We now have membrane-bound organelles, which are these specialized compartments within our cells that can execute new functions, which were basically the necessary infrastructure for all multicellular life. So in other words, this merger, this first biological download, is the reason it's the thing that powers every single life form that we can see today, and it still runs our biology. What you're seeing in this video, I find it extremely exciting. This is why I started working on mitochondria in the first place. But this is happening inside your bodies right now as we speak. Your cells are reaching out to each other, forming microscopic connections, physically uploading and downloading mitochondria between them. This is just astonishing. It happens in our neurons, in our stem cells, in our immune system, and even in cancer. And while I believe most of us have heard the term mitochondria are the powerhouse of the cell, they're actually much, much more than that. Yes, they do create 80% of cellular energy, but they're not just the powerhouse. They are the control room. They generate the necessary precursors, the materials that our cells need in order to stay alive and do their different functions. And they command some of the most important cellular decisions that our cells make on a daily basis. So from our neurons forming new connections, our muscles contracting, and even the final call for cell death is made by our mitochondria. Yet after decades of trying, we have still not been able to engineer them. And the problem is actually mechanical. So we're getting a little bit into the weeds here, but over those billion years of evolution, our mitochondria actually lost all of their ways to import DNA from the environment. And all the other DNA delivery technologies that we have, they rely on our ability to punch holes through the membrane, which is basically a non-starter because it would cause both our mitochondria and our cells to die. So what we're doing is we're taking a completely different approach. We're actually trying to disguise mitochondria as the bacteria that they once used to be, and then using viruses to target those. And for this to work, two things have to be true. The first is we need to be able to deliver those bacteria-recognizing viruses called bacteriophages into our cells. And the second is we need to be able to disguise our mitochondria as their target. And in our preliminary data, we've shown that we can do both of these. Where does that lead us? In the short term, we want to solve mitochondrial diseases. These are rare diseases, about 5,000 patients in the US, and there is no cure. Median survival is 5 years. However, mitochondria have such a central role in our physiology. This modality could actually be used not just for mitochondrial disease, but in plenty of other systems, because mitochondrial dysfunction today is recognized as one of the core hallmarks of aging. So this relates to neurodegenerative diseases, muscular diseases, hematopoietic diseases, all the ones that I mentioned in the beginning. And actually, after this, we want to use mitochondria as a platform for integrating new things into our biology, as a platform for human health and human performance. So after we managed to create this viral retargeting platform and develop large genome language models to create synthetic mitochondrial genomes, we're going to use mitochondria as a platform for downloading new functions into them. So that means using them to improve synaptic plasticity, baseline neuron firing rate, even potentially using them to create radiation shields for space health, or integrating non-biological chemistries or bioorthogonal chemistries into our cells. All of this hinges on our ability to be able to engineer mitochondria, and this is exactly what we're doing. We have a 5-year program for this, and we have started symbiosis labs in order to execute this mission, turning mitochondria into a platform for human health and performance, changing the cell's building blocks and architecture. We are very excited to be in late-stage negotiations with a government agency for an $8 million sponsored research agreement. And our team is built from experts in both bioengineering from Harvard and MIT, mitochondrial biology experts, and delivery specialists. And we are seeking $4 million to be able to develop this platform that can traverse the cells in our body, sense, integrate, and change them with molecular precision, but whole body scale. Thank you very much.